Hey guys, so we're going to talk about all the different words for DNA and chromosomes and chromatids and chromatin and how those all kind of differ from each other. Excuse me. We use these certain terms during like really certain times in the cell cycle. And so we're just going to kind of go over like when to use these different terminologies. So let's just find your next page. Let's call it chromosome structure or DNA term. Let's do DNA terminology. Okay. So essentially what we've gone over so far, let's kind of flip back, is we did go over the structure of DNA. And the way that we've been drawing DNA so far is this linear structure where it's a double helix, but it's still in a linear string of DNA. And we did that same thing over here when we were talking about DNA replication. So that type of DNA, when it's unwound, it's called chromatin. So we're going to kind of draw the cell in different stages of the cell cycle. And then I'll go over what we call the DNA during those different times. So let's just first start with a cell. And then in that cell, so let's just label, this is our cell, so that's my membrane. And then we're gonna have a nucleus within this cell. Cute. So when a cell is non-dividing, so when a cell is in G0, or if it's in G1, S, or G2, so if it's in interphase, the DNA is what we call uncondensed. So let's just say, so this is a non-dividing cell. And when we say non-dividing, all we mean is it's just like not actively in the M phase. Which is most of our cells most of the time, right? So like the M phase is one of the shorter phases. It takes about 24 hours. So if we were to like just grab a cell and look at its DNA, more likely it's going to be uncondensed than it will be condensed. So in a non-dividing cell, that's not, meaning it's not in the M phase, the DNA is referred to as chromatin. And what chromatin is, I'm gonna zoom in. Chromatin is uncondensed DNA. Oh, there's a ching around. And it's going to be wrapped around. Oh, here she comes, guest appearance. Hi, what are you doing? Are you making your guest appearance? Yes? You're such a weirdo. Okay. Okay. Oh, there she is. All right, she's going to flop again. Flop. Okay, we're just gonna... Oh my gosh, so many things happening, excuse me. So chromatin is uncondensed DNA, and it's slightly wrapped up or slightly coiled. And what it's coiled around, excuse me, it's coiled around these proteins, and these proteins are called histones. I'm just gonna leave the cat there and see how this goes. Okay, so essentially what it looks like is it's not just like strands of chromatin, because you have enough DNA in your genome and it's about like 3 meters long. Okay, so in every single cell of your body except for your sperm and eggs, you have 3 meters of DNA in there that will not fit inside these microscopic nuclei. So we have to take those approximately 3 meters of DNA, and we have to start like wrapping that DNA around these small proteins, and that kind of helps to shorten and condense the DNA. So we're gonna have like slightly condensed, we still call it uncondensed because it's not as dense as a chromosome. But essentially, it would look like this. So histone proteins are shaped like little barrels. Let's draw a couple of histones. So these are histones. It's a barrel-shaped protein. And what the DNA does is the DNA will coil lightly around the histone. So I'm just going to draw my DNA as a single strand, but it'll kind of approach the histone, and then it coils around it. And then it'll go to the next histone and coil around it. And so we're taking this like really long strand of DNA and we're condensing it into just a shorter strand so that they can all fit inside the nucleus. So we're going to kind of separate our maternal and our paternal DNA. So you're just going to need two colors that you can distinguish between. So I'm going to just make a little key up here. I'm going to have these two colors. This color is going to represent my maternal genome, meaning this is all the DNA you inherited from your mother, your biological mother. And then I'm going to use this color to represent paternal DNA, which you inherited from your biological father. So you kind of heard like it's 50-50. It's approximately 50-50. You can definitely have more paternal than maternal DNA, just how meiosis works. And so we'll talk about that in the next video. So in a non-dividing cell, it's called chromatin. It's uncondensed. We say that it's a spaghetti-like in structure because the DNA is not tightly wound. And so in your nucleus, you will have 46 little pieces of this chromatin spaghetti-like structure. So you'll have 23 pieces of paternal, and there'll be various lengths. So you'll have some that are longer, some that are shorter. We're just going to draw a few. And you'll have 23 maternal little pieces that are similar in length. And they're all just kind of like jumbled up and messy in the nucleus. So when you look at a cell that is in interphase or G0, the nucleus appears really, really dark because it's full of all this chromatin. So let's just kind of go over numbers. In a regular human cell, you're going to have 46 pieces of chromatin. You have 46 chromosomes. Oh my God, she's so cute. Look at her face. You have 46 pieces of DNA. You get 23 from your mother, 23 from your father. Okay? So total, we have 46 chromosomes. You'll have 46 separate pieces of chromatin. Okay? Oh, she's clicking stuff on my computer with her booty. Okay. So let's just kind of flesh that out. You inherit 23 from your mother. We're going to use our symbols here. So this means, that's father. That's fine. 23 from your father, and then you get 23 from your mother. Those are just the symbols for man and woman. Cool. So when the cell prepares to divide, so we're going to draw our cell in a different stage now. So when the cell goes into prophase, which is the first step of mitosis or meiosis, so this is a cell preparing to divide. Also known as prophase. So the first step of the M phase, mitosis or meiosis, the DNA will condense. So instead of having chromatin like this, that's super spaghetti-like, you're actually going to have an event called supercoiling. And what supercoiling does is it takes this DNA wrapped around histones, and then it starts to take this and coil that. It'll take that and coil that and coil and coil and coil and coil. So it's like coils within coils within coils of DNA. And it's taking these long strands of chromatin and it's condensing them down into these more dense chromosomes. So let's draw what the cell would look like. So we're going to have, from now on, we're just going to draw the nucleus. So I'm just going to be drawing this purple circle here. Just make sure you understand, like, there's a cell, but I want to draw the nuclei kind of big just so you can see what's going on. And I'll label it as the nucleus. Okay. So a few things are happening during prophase. First, your chromosomes are going to condense, and then your nuclear envelope is going to dissolve. So this nucleus will not be here for the entirety of prophase. I'm going to draw it here just so you can kind of orient yourselves. But when a cell is prepared to divide, one of the things that prophase does is it takes each of these small pieces of chromatin and it condenses them into a chromosome. So where once I had the super long string, I'm going to coil that string, and now I get this condensed little chromosome. And so you'll have chromosomes inherited from your mother, chromosomes inherited from your father. Some will be pretty short, and then some will be a little bit longer. Okay, so those are like 4 little pieces of chromatin that are now condensed into chromosomes. So numbers-wise, we had 46 pieces of chromatin. You're going to have 46 chromosomes. You'd think I didn't give her attention like all day. Goodness gracious. You have 46 chromosomes, sorry. Once again, you have 23 from your father, 23 from your mother. Okay. So let's kind of go over how we refer to chromosomes. Okay. So in a somatic cell or a body cell, there's a body cell. So anything that's not a sperm or an egg cell. All somatic cells have 46 chromosomes. Okay. Meaning you have what we call 23 homologous pairs. Okay, so how we say you have 23 from your father, 23 from your mother, they're paired up in pairs. Oh, so redundant. And the pairs are called homologs. So remember, homo means same. So a homologous pair of chromosomes are chromosomes that code for the same genes. They might not carry the same physical characteristic, but they carry the same gene. For example, when you look at chromosomes under the microscope, they have this distinct banding pattern. So you'll actually see like different striping patterns on the chromosomes. One from your father and then the one from your mother will have similar banding pattern. And so we can kind of figure out, it's like this one's from your father, this one's from your mother. We can figure out that these 2 chromosomes, while they come from different people, they carry the same trait. Meaning, this top banding might be the gene that codes for your chin. So whether you have a cleft chin or not a cleft chin. And then this next trait might code for the shape of your thumb. Whether you have a straight thumb or a hitchhiker's thumb. And then this next trait might code for your hairline. Whether you have a straight hairline or a widow's peak. Okay? So they're going to carry the same genes in roughly the same order, but they might not carry the same, it's called an allele, which is a variation of a gene, right? So while both your mother and father have the gene for your chin shape, your father may carry the cleft chin and your mother may carry the non-cleft chin, and then the combination together will give rise to what kind of chin you have. So we pair up these chromosomes based on a few things. We pair them up based on their length, because if they carry the same amount of genes, they're going to be approximately the same length. And then we also pair them up based on their banding pattern, because that's showing the density of DNA in different spots. So these 2 right here, we would call a homologous pair. Okay, and each of these are made of a chromosome. So we have 1, 2, 3, 4 chromosomes in my nucleus in total here. And like these two are similar lengths, so they might have like a similar banding pattern, so they could also possibly be homologs. When we talk about DNA, when we organize it, we create these structures called karyotypes. And what a karyotype is, it just takes all the chromosomes in a cell and it organizes them from chromosome number 1 through chromosome number 23. So like, I'll show you, it's a little chart of chromosomes, but it organizes them from number 1 to number 22. And then it goes, longest chromosomes are number 1, the shortest chromosomes are 22, and then the last pair on a karyotype are your X's and your Y's. So you'll either have XX or XY. So when you're looking at a karyotype, let me not mess this up like I did last time. Oh, I did it. Okay, so when you look at a karyotype, they look like this. So you can see that we organize them from left to right in rows, and you number them. So we have chromosomes number 1, you can see the banding pattern. So this is one from mother, one from father, all the way through chromosomes 22, and they're the shortest of our chromosomes. And then you'll have the sex chromosomes here, and so you can tell that this individual was born a female, or sorry, male, because they are X, Y in genome. So cool. So those are all homologous pairs. Oh, now we're gonna put this back and not mess it up. Yeah, we did. Okay. Next, after we kind of exit the M phase, go through G1, and then get back into the S phase. So we're gonna draw a cell that's gonna be after replication. So after S phase. I should have done that backwards, but that's okay. You will have, everything will be doubled because we've replicated everything. So let's draw our nucleus. Bless you. Oh my God. Ching, you are making this video everything. Okay, so after replication, what we have, instead of having singular chromosomes, they're all gonna start looking like little X's, okay? So where we had a single chromosome, like so, it's now been replicated after the S phase. And so it will actually have 2, okay? So I draw them like little K's just because it makes it easy. So the other one that came from the father will also be replicated. So when you see a replicated genome or a replicated karyotype, all the chromosomes look like these little X's, okay? They'll have the same banding pattern because they should be exact copies of each other. If there was a mutation, they may be different, right? So remember, like these 2 original ones had similar banding. And so now their copies will have similar banding to those originals. Okay, bye. She's over it. She's over us. Cool. So these are 2 of the same chromosome from your father. These are 2 of the same chromosome from your mother. They're held together at a certain point. It's not always like in the center, like I drew it here, but that could be like held together down here. But there's a kind of system of proteins that'll hold, we call it like the waist of the chromosome together. And it's referred to as the centromere. It's a system of proteins. During mitosis, those proteins will get pulled apart. So we have now replicated DNA. So now our language gets a little different, okay? We still have 46 chromosomes. Okay, you have 23 from your father. You have 23 from your mother. Okay. If you look here, it does appear like everything was doubled. The reason that we don't double our chromosomes is because when they're connected like this, they're referred to as one chromosome. Okay, so this is one chromosome. This is another chromosome. So in this nucleus, we have 2 chromosomes total. Because we have replicates, now we have 2 of something else. So this chromosome is made up of 2 sister chromatids. Okay, so we have another new word here. These 2 together, we still refer to them as a homologous pair because you still have the one from your mom, one from your dad coding for the same genes. But now each homologous, the chromosomes within the homologous pair are made up of 2 sister chromatids. So a sister chromatid is just a replicate of a chromosome. So they should be identical, but they might be different. So in this diagram, while we have total 46 chromosomes, you would have 92 chromatids because you have 92 of those copies. Okay, and then once we get over here, so sorry, this cell looks like this going into prophase. So like we should have drawn these as duplicates. I made a boo-boo. So the cell looks like this going into prophase because after anaphase, once they get pulled apart, so this will be during or after anaphase. And if you don't know the steps to mitosis and meiosis, watch my next video. Those homologous pairs will get split up and the chromatids will get split up. Okay, here's my last nucleus. So at the end of anaphase, what starts to happen is all of these chromatids have been pulled apart. So one chromatid will go to one cell, the other will go to the other cell. One will go to one cell, the other will go to the other cell. So what ends up happening is you end up with the same amount of chromosomes as you started with. So on one side of the cell, we call them poles. You'll have one of the chromosomes, now called a chromosome. Over here, you'll have the other. So before they were connected by the centromere, after anaphase, the centromere gets pulled apart and one chromatid is here, one chromatid is here. Because they're now separate from each other, they're not bound to each other anymore, they're referred to as chromosomes again. So you only have chromatids when they are bound to each other by a centromere. Once they get pulled apart and they're separate from each other, they're now called chromosomes. And then the one that came from your father will be separated as well. And then eventually after telophase, the nucleus will reform and then you'll kind of have separate cells. So one cell will have the original 2, the other cell will have the other 2. Cool. So remember this one came from mother, mother, father, or father. Okay. So here you'll end up still having in each cell, so in each new cell, there will be 46 chromosomes. Okay, 23 from your mother, 23 from your father. So you'll have 23 different ones of these, 23 different ones of each of these in your new cells. The existence of chromatids is no more, so none of these are referred to as chromatids. The only time you have chromatids is after S phase, so only between like the end of S phase and anaphase do you have chromatids. Interphase and the other phases like telophase and cytokinesis, everything is referred to as a chromosome. Okay, that's it for DNA terminology. Come back and watch my mitosis meiosis video. All right, bye guys.